Hi, everyone. Thanks for coming today. My name's Riley Juenemann, and I'm a senior in undergrad, double majoring in math and computer science at Tulane University. And today I'm going to be sharing some work on developing a dashboard of statistical tools for categorizing different types of particle movement inside of cells. And this was done with Professor Scott McKinley at Tulane and Professor Christine Payne at Duke University. So we're going to start with a tiny bit of biology background, and this is a diagram of some of the mechanisms that play a role in transport inside of cells. And to demystify some of the language a little bit, so cargo, which we're using to refer to things like organelles, nanoparticles, or vesicles inside the cell, is what we see during the experiments. And that cargo is being carried by these proteins called molecular motors that transport the cargo along these sort of cellular highways, which are called microtubules. And there are particular motors that move in particular directions, which further adds to the complexity of this biological context. And there's typically more than one motor attached to the cargo too. But of course, we can't see that. All we're seeing is the cargo. And so the way that we see this cargo is through experiments such as fluorescence microscopy. And these can help us sort of take a glimpse at those dynamics within cells, although we don't understand them very well just yet. And scientists get hundreds of time series trajectories as outputs of each one of these fluorescence microscopy experiments. You can see it's a series of images where you have these like little glowing dots, and then we connect them together to form the trajectories. And because they get all these data from these experiments, there's a lot of data that needs to be analyzed and processed. And it's kind of a pain to have to look at this all sort of qualitatively. And so our goal is to develop a tool that can be used to automatically categorize different types of particle movement in cells. As you can see by the plots I have here, there's great heterogeneity between paths. The paths look a lot different. And some of the paths are easier to categorize than others. So here are a few of the easy movement type categories. We have these directed particles, which you can think of as like moving in a particular direction with a purpose. So they know where they're going, and they're going there a lot faster than some of the other particles are going places. And so here the cargo is being transported by that molecular motor protein. It's being carried along the cellular highway, which is the microtubule. And it might look something like this for this particle. Anchored particles are also attached to that molecular motor. But in this case, that molecular motor is not moving anywhere. It's staying in one place. So it's still tethered to a fixed point and fluctuating around that. And then finally, freely diffusing particles are not attached and are moving in a Brownian motion pattern based on collisions with other particles or the fluid inside the cell. And since biological data is messy, we also see some paths that are harder to categorize. So this top one looks like it's moving back and forth and back and forth and back and forth, or pacing. But after talking to our collaborator, we recognize that this one is actually an error in the process of connecting the trajectory from all these little dots. So this one is actually two of these anchored ones, like over here and over here, that just happen to be close enough together that they're being connected by the algorithm. In this bottom plot, there's lots of crazy behavior going on here. It seems to go in one direction and then cycle back and then come back again. So we're not really sure what to make of some of these plots. And so if we had this sort of automated categorization tool, maybe it would allow us to recognize things that we can't observe qualitatively. And one application in particular of this tool would be to aid our collaborator, Professor Christine Payne from Duke University, in determining the impact of titanium dioxide nanoparticles which are found in a lot of consumer products. So things like sunscreen, paints and pigments, food coloring, even toothpaste. And we want to know, when workers are in the factories that are producing these products and breathing in these nanoparticles, what's it doing to their lungs, sort of at a below toxic exposure, but over an extended period of time. And in particular, related to this project, we're interested in the movement patterns of these things called lysosomes, which are kind of like the stomachs of the cell. And when these particles enter the cell, they go into those like cellular stomachs, except for it makes those lysosomes balloon up. So they're like much larger, sometimes like 3 times as large as they would be in normal cells. So we want to understand whether those like super big stomachs in the cell move differently than sort of the standard ones. And so with this problem, goal, and application in mind, the question becomes, how can we use math to categorize these different movement types? And so since we're tackling this large open-ended question, we're going to start by determining the simplest relevant problem. And in this case, it's starting with data that we already sort of know the answers to. We know what the movement types are supposed to be. And so we can simulate some of these paths using something called Langevin dynamics. And so this is actually derived from Newton's second law, force equals mass times acceleration, and represents the different forces that are on the cargo in the cell. And so we can do some rearranging and use some stochastic differential equation notation to arrive at this equation, which we can use to simulate those sort of easy categorizations we were talking about. The free diffusion, where the cargo isn't tied to anything, so it's just randomly fluctuating. The stuck diffusion, where it's anchored to that fixed point. And then directed transport, where the anchor is moving with some velocity. And so those free, anchored, and directed movement types come straight from that stochastic differential equation, but we're also interested in a few other sort of bonus types. If you remember that path that looked like it was moving back and forth and back and forth, we're interested in picking out those paths that might be candidates for that tracker error. We're also interested in subdiffusing paths, which are like Brownian motion with a little bit of memory. And then skating diffusion is also a little bit of a hybrid path. It's going to be free in one direction, but anchored in the other. And so we can choose parameters for our stochastic differential equation to match our target live cell data. So now that we have our simulated data, we can work on defining some statistical features to distinguish between the movement types. And the first thing we do is determine the major and minor axes of the path based upon a method that's similar to principal components analysis. So we call the line with the greatest range of points the major direction. And then the perpendicular line to that is going to be the minor direction. And we take the extent or standard deviation from projecting the observed points onto both directions. And you might ask why this metric is useful for distinguishing between the different types. And as you can see here, the freely diffusing and the anchored diffusing are going to be more evenly distributed along that major and minor direction. So kind of more of like the trajectory itself might be more of like a spherical circular shape, while the directed transport paths are going to have much more spread along that major axis. They're kind of elongated because they're favoring that particular direction. And we can see that in this plot of the standard deviation in the minor axes and the standard deviation in the major axes. And when we're looking at this plot, we're really looking at what's close to this diagonal line, because that means that those two standard deviations are close together. And so for those diffusive movement types, the freely diffusing and the anchored diffusing, we're going to see those fall a little bit closer to that line, while movement types that favor one direction are going to come off the line more. So in particular, the skating, diffusing, and the directed transport ones. We also use that major and minor axes lines for our next two statistical measures, where we're testing the independence of increments. And in particular, one hallmark of free diffusion or Brownian motion is independent increments. So where you came from is not a good predictor of where you're going to go next. And we expect 95% of those freely diffusing paths to fall within this box. You might notice that some of the other types have negative autocorrelation values. And this is because that anchor, that molecular motor that the particle is attached to, is more likely to pull the particle back towards the anchor. You might also recognize that there is some overlap between the groups here. So specifically red and blue on this plot. And this is why we need multiple statistical measures to separate the types. For our last two statistical measures, we need to estimate the path of the anchor, which is that molecular motor that's attached to the cargo. And we can do this using splines, which preserve the characteristics of the path. And this is important because those microtubules, the cellular highways that we were talking about, can be bent inside of the cell. And so once we have estimated that parametrized anchor path, shown in red here, we can take the average of the square distances for each time step for each path. And we call this delta anchor. Again, we're going to look at what that looks like for our easy movement types. Since we're using the parametrized spline in our computation of distance, these diffusive movement types are going to have a greater deviation from the spline than the directed transport paths, which are going to follow the spline much more closely. Our final statistical measure is effective diffusivity, which you can think of as a measure of how much space is explored. And this comes from using maximum likelihood estimation. So this says from the data, what is the most likely value that we would see for diffusivity? And we plot this with our delta anchor method, which we were just talking about with the splines, to show the separation with these two statistical measures. Notice that in this plot, those red and blue groups that were overlapping on the last plot aren't overlapping anymore. But now some of the other groups have become much closer together. And again, this is just reiterating why we need so many different statistical measures. And since we can't see in 6 dimensions, we need to use some machine learning. So initially, we plug these final 6 statistical measures to k-means clustering, which is an unsupervised machine learning algorithm that partitions a data set into k clusters in a way that minimizes variation. So it's looking at the ensemble of trajectories as a whole. And here k does have to be specified by the user, although there are a few different sort of like numerical methods that you can use to pick out a good value for k. And for this particular set of parameters that we plugged into that stochastic differential equation to simulate, we have 25 of each of the 6 movement types, and we're seeing all correct categorization. So this is really promising that it's doing so well on the simulated data. And just as a note, this plot doesn't mean too much to us quantitatively. It's plotting based upon some principal components, but it is a nice representation of how the paths are clustered based on distance, projecting that 6-dimensional space onto 2 dimensions. And so given data sets, this is an automated categorization system to help identify cells with more directed transport and cells with more diffusive transport. And I focused on the methods today since this is a math conference, but we've also applied this dashboard to data sets from our collaborators. And currently, I am working on improving upon this work in both my mathematics honors thesis and my computer science capstone, which I'd like to talk about a little bit. For my math honors thesis, we're looking at model selection for diffusive movement patterns. In particular, we're asking the question of, can these methods, which is better at distinguishing between that free diffusion and that anchored diffusion. And also asking the question of, is it easier for free diffusion to like seem like anchored diffusion, or is it easier for anchored diffusion to see like free diffusion? And so as you can imagine, with different like path lengths and things, there is some idea there. Like if you look at a freely diffusing particle for a very small period of time, then it could seem like one of those anchored particles because it hasn't had enough time to really be random and explore places. But mathematically, proving that has been a nice challenge that we've been working on. Also, in my computer science capstone, we've been working on using supervised machine learning, in particular support vector machines, instead of k-means. And for those of you who are familiar with k-means or machine learning, you were probably asking, okay, so you have simulated labeled data. Why are you using an unsupervised clustering algorithm and not capitalizing on the fact that you have those labels? And so that's something that I've also been working on addressing this year through my computer science capstone, is I'm using that simulated labeled data to sort of train a machine learning algorithm. So that way, after we've trained it on a wide parameter space, then we can use that algorithm on the experimental data to get results. So I've been working on that all year. It's pretty exciting. And we're almost there in applying it to some biological data sets. So that's been pretty cool. I also wanted to include a slide, particularly for the students in the room. I know that when I started undergrad, I didn't really have an idea of what research looked like, especially in mathematics. And it's been a really rewarding experience for me in my undergraduate education. And it's allowed me to sort of like set my sights on pursuing this more long-term and going to grad school in applied math. And in particular, there are some really interesting problems in math bio, even if you think, oh, well, I'm not like a bio major or minor. There are a lot of projects that need mathematicians and people with the sort of quantitative analysis tools to work on these large, complex data sets. And so in particular, this project involved things like modeling, differential equations, probability, sampling from distributions from different parameter spaces, statistics, the statistical measures, processing lots of data. When you start a project, it seems like you always spend more time preparing the data into a usable format than you sometimes do doing the math on the project. And then also some programming and some machine learning. And a personal soapbox of mine is a lot of people have heard the term machine learning, but maybe don't know that much about it or are scared by it. And so this is one of my favorite XKCD comics, where it talks about how machine learning is a pile of linear algebra, sprinkle in some statistics. So really the backbone behind it is still math. So thanks again for coming to my talk. Thank you to everyone involved with Math for All, from the organizers to the volunteers to the attendees. Thanks to Tulane and my research advisors. And I'm happy to answer any questions. I should mention this image was actually from the summer I spent in the lab with my collaborator, learning how to culture cells and do my own fluorescence microscopy experiments.